So hi everyone, I'm Dr. Brown from Annapolis University, and I'm sitting today with Dr. Daniel Ashlock from the University of Guelph. Dan, do you want to give maybe a short description of your background?
Hello, welcome from Canada. I am a mathematician trained in abstract algebra and then retrained in helping biologists. I'm a founding member of the Bioinformatics program at Iowa State University, which was one of the first such programs in the world. I'm currently the holder of a Bioinformatics Chair at the University of Guelph. I have publications on epidemiology, though not many. On the other hand, one of them won a Best Conference Paper Award at the Congress on Evolutionary Computation. I actually know more biology than most biology professors, but I know far less biology in any one biology professor's specialty, which puts me in a weird sort of place. Most of what I do is help biologists with their math, and as will probably surprise none of you, I've been spending a lot of time concentrating on epidemiology and the COVID-19 virus, SARS-CoV-2. Okay, back to you, Joey.
Okay, so the idea mostly of this is in order to give you guys a chance to ask any questions. So you can put those through the chat, and I'll basically relay them on so that way everybody on the recording can also hear anything that you want to ask about this. I did already get one of the messages from the Dean, Giancarlo Succi. He was wondering kind of what do you see normally inside of the graphs that are caused inside of this pandemic? So in terms of the distributions that are seen.
Okay, I'm going to come back with, I don't understand the question. Do you mean the contact graphs of individuals, or do you mean the epidemic spread graphs that look exponential?
I think he was mostly questioning about the epidemic spread graphs and the exponentiality.
Okay. What I see right now is that they look exponential in places like the United States that are doing a very bad job of containing it, and in places like China or Korea where they are able to just almost lock everything down, or in the case of Korea, test everyone, they're starting to curve back down. It looks like once this thing really hits a country, if they're trying, they can get it to curve back down after 3 weeks. We don't know how long it'll take to start seriously dropping, as opposed to not increasing so fast, which is what I mean by curve down. The epidemic spread models, though, I mean, the real message in these curves is stay home, reduce contact, only absolutely necessary trips, things like that. Over.
Okay, and so if you were to put this into kind of a mathematical function, what would you expect to see in a normal disease?
Okay, I'm not sure what a normal disease is. And in fact, let's start there. This is not a normal disease. In particular, with viral diseases, usually you become able to give them to other people and you show symptoms very close together. With COVID-19, the symptoms show up 3 to 6 days after you become able to infect other people. This means this virus spreads far more effectively than most, making it exceptionally dangerous, except that its death rate is somewhere between 0.5% and 6%. And I don't mean we don't know where it is in that range. I mean, that's the range we've observed given different countries' measures. In small countries with no real medical system, it gets up to around 6%. South Korea seems to be keeping it near 0.5%, but they're able to treat people immediately. I would guess that in the United States, it will be about 3%, and Russia, which I see from this morning's news is just starting on their infection, can probably also contain it within 3%, and well below that if people will obey the social distancing directions. Joey, did that roughly answer your question, or do you want to ask it again? Over.
Well, I think so, but I think part of this is people are seeing this as being kind of like a normal flu. In terms of, you get it, it moves through the population, and it kind of tapers off, coming up to a kind of a carrying capacity off of this. So are we going to see something along that with the graph, or is this going to keep like exponential for a longer period of time?
Okay, at the current rate of spread, it cannot go exponential for a much longer time because we'll run out of people. Starting with that cheerful thought, this is nothing like a normal flu. It's a coronavirus, and so that means it has a very different pattern than the flu. The most common pattern for a coronavirus is for it to die back as the weather warms and the humidity increases, and then to return again in the fall when it cools off. This is actually quite likely to happen. Also, a coronavirus is an RNA virus. So there are two sorts of viruses, ones that store their information in DNA, ones that store their information in RNA. And without going into any more molecular biology, the RNA ones are very bad at copying themselves. This has the unfortunate side effect that they mutate quickly. And so the reason we don't have a cure for the common cold is that there is no such thing as the common cold. There's 1,000 different common colds that can turn into one another by mutation. And so anything we worked out to kill it, it would probably escape from. It looks like the coronavirus has a lower mutation rate than the common cold, or to be more accurate, than the rhinoviruses that cause most common colds. It's a coronavirus, and while coronaviruses do cause colds, they don't escape from the immune system as fast as rhinoviruses. So that means that we probably can develop a vaccine, but we're going to see it popping up again and again as people release the quarantine or the social distancing measures too soon. I note, for instance, that President Trump has said he would like to release the social distancing measures on Easter in about 3 weeks because it's such a beautiful date. Pretty much every health official in the United States has then returned by saying, no, no, don't do it. No, 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 don't do that. No. But we'll have to see what happens. The other problem is different coronaviruses have different rates of having a second wave, and we have no data on the second wave of this one beyond the fact that there are supposed to be 6 cases of a person catching it twice. However, at that level, it may simply be statistical error. We need more data. Over.
I have one coming off of that, and that is our immune system better prepared for a second or third infection? Does that have anything to do with the protections from this?
Yes. If you catch the virus a second time, your chance of having an awful outcome, like have to go to the hospital and have your lungs pumped out, is lower, but it doesn't drop to 0. So the second wave hits people that have already been infected less hard in other coronaviruses. For this one, okay, that's probably how it's going to work, but we don't know because it has never happened before.
Okay. And so how does the development of the virus depend upon the amount of initial contact with infected people?
This is actually a classical piece of epidemiology that was discovered among fig wasps that prey on fig trees in Central America, but I think it applies to the virus. If the virus's ability to get to the next host is enhanced, if it's easy to pass the virus along, that favors viruses that make lots of copies themselves, do enormous damage to their host, and in essence are more deadly. If, on the other hand, it's hard for the virus to get to the next host, then the evolution of the virus favors the ones that don't do a lot of damage to their host. So a secondary effect of maintaining quarantine and social distancing is you will be encouraging the weaker strains of the virus by evolution.
Okay, so I've got one question here. How did this virus first appear? And do you think that there's human intervention? Like, is it a human-made? Is it a bioweapon?
Okay, so the short answer is no, it's not a bioweapon. And then here's the supporting data. First of all, it's a terrible bioweapon. It doesn't kill people enough. It spreads too fast and in too uncontrolled a fashion. A bioweapon would not have been made with this feature of being infective for 3 to 6 days before their symptoms. And it's also not killing people efficiently enough. All this thing does is disrupt economies, and it seems to disrupt all of them. So that's sort of level one, is this a bioweapon? Level two, is this a bioweapon, is that a research team actually went through about 5 different sequenced coronaviruses and looked for the fingerprints of all of the different sort of genetic engineering and DNA surgery techniques or RNA surgery techniques we know. And there is no evidence this virus was synthesized or created from an existing wild virus by technology. People checked. It bothers me a little that we took a research team that could have been working on something useful and went and checked to see if this is a weapon. I note right now that the Chinese information people are claiming that it was a US-made weapon, and they got the idea from a Canadian conspiracy theory website, and that a number of people in the US government are claiming it was made in a secret Chinese lab. None of these people have produced any evidence. Here's the third line of evidence. This coronavirus is very similar to coronaviruses that appear in bats and pangolin and snakes, all of which are eaten in China and often eaten raw for medicinal purposes. This means that there's another source for the virus besides a bioweapon lab, and that's natural reservoirs. Now, the other problem is this thing resembles viruses from all 3 of these species, and the current theory is that it is a snake virus recently acquired from a snake eating a bat that got into people because somebody was eating undercooked snake soup. I would give this theory about a 3% chance of being right, but I know of no theory with a higher chance of being right. We really don't know where this virus came from. We just know that new viruses are showing up all the time, 5 or 6 per year. This one we noticed because it's killing people. Over.
So what is the current prognosis for research in terms of a vaccine in the near future?
Okay. Classical vaccines, we isolate antigens either from antibodies of infected people or possibly directly from proteins produced by the virus. It takes about 1.5 years to get one of those up and running. When the SARS virus came rolling through in the early 2000s, it took 5 years. So we've gotten a lot better. I'm sure we will have vaccines in the pipeline to appear in 18 months. But there is a unicorn, by which I mean a $1 billion funded startup company that has been looking at making vaccines directly from RNA. They claim they will have a COVID-19 virus out in 6 months. And their claim seems plausible based on what they have published on their methods. Beyond that, the World Health Organization just started a trial in 85 countries of drugs that are not vaccines, but which reduce the incidence of severe cases of COVID-19. And the Netherlands has just started a trial of a 100-year-old tuberculosis vaccine. Now, that sounds a little nuts at first because tuberculosis is caused by a mycobacterium, not a virus. But the people who are doing this have been working for 10 years and have a series of papers that shows that this particular inoculant causes general immunity to everything, including viruses, for about 1 year. It's not very good immunity. It gives you about 30%. But they're going to be trying it on 1,000 healthcare workers in the Netherlands starting next week. And if this happens, it'll reduce the rate of people getting sick. And then finally, I've seen at least 12 different scientific research groups looking at old ideas and trying to bring them up. So I would say, I'm sure we'll have a vaccine in 18 months. We may have a vaccine in 6 months. We may have effective treatments, not vaccines, but treatments that make the virus do less damage in a month or 2. And also, oddly, we already have a at least approved in the US and known to be effective technique for saving people. The problem is you need to centrifuge the blood of a survivor down to blood plasma containing antibodies and transfuse large amounts of it. So this can only be used on a relatively small number of people. So that's what I know about the current vaccination. And I'd say just keep an eye on the science news because we've never been trying this hard before on anything. This is bigger than the American atomic bomb project during the Second World War, and it's spread out over dozens of scientifically literate countries. Over.
Okay, so the other thing that I heard there was bacteria, virus. What exactly is the difference?
Bacteria are complete cells. They have a nucleus, they have protein manufacturing machinery, and they're independent living organisms. They need to eat, but they're small. There are also two sorts of them. The ones that are like us, the ones with cell nuclei, and then things like mitochondria. And then there are simpler bacteria that have collections of DNA in them, but they're self-replicating organisms. Those are bacteria. Viruses are genetic parasites. They cannot reproduce themselves. A virus injects itself into a living cell, and keep in mind, it's tens of thousands of times smaller than the cell it's infecting. The virus then hijacks the cell's DNA replication, RNA replication, and protein-making machinery to make viruses. It turns out that the human genome is about 8 to 12% viruses that infected a human being and didn't succeed in causing a disease and got integrated into their genome. And so viruses are ancient. New viruses form out of human genetic material occasionally. There's one called Phoenix that reappears in human beings once in a while and also appears in chimpanzees once in a while, and it's the same virus. So it's 5 million years old, and it just occasionally starts running again in our cells, but not at a level that does any damage. So yeah, that's the difference. Viruses are super small genetic parasites that hijack your cells to make more copies of themselves. Bacteria are independent parasites, single celled. And in fact, one of the things that's sort of interesting is we may be able to take out a lot of bacterial diseases with viruses. There's a type of virus called a bacteriophage that eats bacteria. And it was pioneered as a medical treatment in Russia in 1915, where they found a couple types of cancer that could be cured with a bacteriophage infection. But then people figured out what viruses were and got very scared of using infections agents as medicine and stopped doing research on it. We're going to have to pivot back to that. And so in a way, we need to study viruses a lot more, not just because of things like COVID-19 killing millions of people by the time this is over, but also because viruses are just manipulatable genetic machinery. The bacteriophages turn out to be incredibly host specific. After a lot of testing recently, we found that they never jump and turn into human diseases. And by never, I mean not in any of the experiments. Over.
Okay, so two that are kind of bridging into one another. What's the probability of the virus now mutating, gathering heavier symptoms or a higher mortality rate? And off of that, could this lead into the second wave of infection? Could it be longer because of that?
So there I'm going to give you a firm maybe. We don't know. We don't have enough information about how this virus changes. On the other hand, the possibility that a much worse version of it will arise is bigger than zero. And so you want to cut that off right at the beginning by not letting the virus spread at all. If the virus is spreading, the chance a worse version of it will show up is enhanced. How big and how long could the second wave of infection be? Again, we don't know. Almost certainly much smaller than the first wave, not because of anything to do with the virus, but because it won't be taking us by surprise again. We're going to have all of the laws and social measures and customs in place, and these people who are going, oh, I'm young, I won't catch the virus, I'm going to go have a beach party with 100 of my friends, will have had enough of their friends die that they'll be taking this seriously again.
Okay, so why is the situation so bad in Italy? They say that every day in the Russian news, they mostly talk about the situation in Italy.
It's essentially because Italy acted as if nothing was wrong for 2 or 3 weeks and got started late on their prevention measures. It's nothing special about Italy other than, as you may know from having interacted with Italians or read about the Italian government, that they're a bit disorganized. So they started too late and they now have a serious problem. interestingly, one of the highest infection rates in Europe is in Switzerland, and it's because they have a lot of Italian guest workers. So, if you would think anybody could screw down the movement of people and get things organized, it would be the Swiss, but they're having troubles with it because it's coming over the border from Italy. So yeah, nothing special about Italy except they started too late.
Okay. Next question. UK and Netherlands are basically choosing kind of a non-confinement strategy. I mean, UK's now moved to it. There's kind of these ideas of getting 80% of the population through the virus as quickly as possible. Is this a sound strategy?
No, it's the stupidest strategy anyone has come up with in the world. It is based on misunderstanding epidemiology. This is what happens when a politician has somebody explain a concept to him and doesn't understand it. So what they're calling this is herd immunity. I note that Boris Johnson, the Prime Minister of England, has tested positive for COVID-19, which is just sort of a sidelight. And a couple of days ago, his country did an abrupt about-face and decided not to use this herd immunity strategy. I believe the Netherlands is changing its mind too, but I don't know. Most of what I hear is from an Italian colleague of mine who moved to the Netherlands about 6 months ago. And he is very angry, and his wife is very angry in Italian. So I think they're having trouble persuading them. But let me explain what herd immunity is, and from that, why this is a deeply stupid strategy. The idea of herd immunity is simply modifying a population so that the number of people infected from somebody that has the disease is less than 1. The number of people a sick person infects is called the R value for the epidemic. If the R value is less than 1, it dies out. Okay. Now, herd immunity is used, the term is used in the context of vaccinating people. And so once you know something about a disease, you can figure out how many people you have to vaccinate or what fraction of the population you have to vaccinate in order to get herd immunity. And somehow Boris Johnson's government heard about this and went, oh, that's great, because then we'll be past it and it'll be over. The problem is that they would have had to kill 2 million people to do that. If you have a vaccine and you get enough people to vaccinate to grant you herd immunity, that's minimizing the number of deaths. A herd immunity strategy without a vaccine maximizes the number of deaths. There is no strategy that will kill more people than that. That's why I say, no, it's a stupid strategy. And the problem is they didn't understand that an epidemiologist would only talk about herd immunity if he had a safe, effective vaccine already in his hand. Over.
Yeah, I think that's basically herd immunity by how we did herd immunity back in the Black Plague.
Here's an interesting sidelight. The Black Plague isn't a serious disease anymore. That's because everybody alive today in Asia and Europe and most of Africa is descended from people that survived it. So we still have herd resistance to Black Plague. If it breaks out now, it won't be nearly as bad as it was then. But look at the price they paid. The mortality was 1/3 of all human beings in Europe, and in some places it was up to 80%, in densely packed cities without sewer systems. Over.
So thank you very much for that answer, was their response back. I don't have any questions currently at the moment, but let's maybe I'll give one of my own, and that is, what can people kind of do right now? What should people be doing? Because I mean, there's a lot of miscommunication and news about this. What actually, from your perspective, should people be doing to protect themselves, their loved ones? What action should they make?
So step 1, don't go out unless you have to. That's to pick up medicine, pick up necessary cleaning supplies, pick up food. One thing I'm doing is I used to go shopping 4 or 5 times a week because I walked past the grocery store on my way home from work. Now I'm going shopping about once every 10 days with an organized shopping list. If you can get your doctor or pharmacy to give you routine medication in a larger supply so you have to go back again less often, do it. In Canada, they told us, stock up on all your medications. I went to my pharmacist, and my pharmacist said, oh, wait, you're not, oh, right, COVID-19. Yes, here you go. Here's your refill on your high blood pressure medication and your diabetes medication. And so I'm running low on my current supply, but I have a bag full of the next batch of it, and that's good. Do not even have a few friends over. Like I said earlier, the virus is infectious before it causes symptoms. Meet online, meet virtually. I talked to my neighbors from 8 feet away, and here's another thing. I made a deal with my neighbors. Anybody gets sick, the rest of us will do their shopping for them and leave it on the porch and settle up after the epidemic is over. It's not a big worry. Don't hoard things. Right now, it's very hard to get toilet paper in Canada. We just by dumb luck shop in bulk and happen to buy one large package of it just before this all hit. And so we're not having trouble, but there are huge problems, particularly the elderly will go to a store and have things they need and they're all stripped off the shelves. So don't do that, please. Remember to keep eating well and remember to exercise. Exercising will keep you healthier. It'll keep your immune system better. And what my wife and I do is we go for a walk in the morning. And if we see somebody else, we walk on the other side of the street or, on somebody's lawn around them. So don't get close to other people, but don't stop exercising. The other thing is vitamin D will boost your immune system a little. So if you can take vitamin D, eat liver, or spend time with your skin in the sun, those help a little. But mostly just don't go out, don't do things that might spread the virus. The other thing is this thing about hand washing is really serious. Normally you would think an alcohol wipe for your hands would be better than soap, but it's not, because the virus can laugh at alcohol. It's in a pretty armored capsid that protects it. So it might rinse the virus off and it'll kill bacteria, which is good. But soap is a surfactant. It clings to both water and the virus's surface. And so hot water and soap and rub your hands will actually break the viral particles apart and kill them. So washing your hands with soap is good. And when do you wash your hands? People are saying often. I wish they'd be more specific. Right after you pick up something off your porch that somebody delivered. Open it, shake it out of the package, throw away the package, and then wash your hands. Also, wipe off your doorknob so that the next guy, that uses the doorknob doesn't get killed. Anything you pick up and put down a lot, your phone is a wonderful example, keep that clean. Wipe it off with, a damp cloth fairly often, and then run hot water and soap in the cloth. I think that's all I know that you can do unless you're a researcher or something. And in that case, help. Over.
Okay. There's been some questions also about vitamin C.
As far as I know, vitamin C does not help much with this virus. It won't hurt. The human body can tolerate huge doses of vitamin C without harm. That's not true about vitamin D, incidentally. You can poison yourself with vitamin D, so don't take extra. But if you want to take vitamin C, there's no particular evidence it helps. It doesn't hurt. It might help, and we just haven't noticed yet.
Yeah, and
I think I see another good question. Oh, go ahead.
Yeah, I think vitamin D also in Russia is a little bit harder to get because they don't spike the milk supply like they do in Canada. Because in Canada, we have the enriched milk and it has the vitamin D in it. I know like over the winters, I get a bit, messed up because of the vitamin D load. And I then started having to take the supplements because they didn't have it in the milk. But there is another question here. Detection is imperfect. Understanding how many people that are infected is not that easy. What would be good indicators for this simulation or situation? Number of actions within intensive therapy blocks. Like how would you actually get a good number and metric for the actual number of cases?
Right now, this is something my wife told me yesterday, which caused me to kind of go sit down on the couch for a minute and go, oh no. It turns out that the current RNA-based PCR test for COVID-19 being used in most countries is 60% accurate. It fails to detect a virus that is present 40% of the time. So yeah, the detection is terrible. It will be getting better as time goes by. And the problem is that it's hard to get a sample containing the virus. Right now, they jam a swab through your nose into your throat. And if that sounds uncomfortable, well, yeah, you're right, it is. And it has a decent chance of not picking up the virus. The PCR test is just fine once you've got a sample with the virus in it. Okay, having said that, there really isn't a good way to get a handle on the number of people with COVID-19 other than looking at dead bodies. The number of people that died of it is a fairly accurate measure of how prevalent the infection is in a country. You know, low in Russia, high in Italy, things like that, somewhere in the middle in Canada. So basically, this is yet another reason that avoiding other people is a great idea, because we don't have a good grip on how many people have this thing. I have a meeting at 10 o'clock with a group of people developing a technique for creating strategies for deploying a vaccine once we have them and issuing quarantine orders until we have them. Okay. And I sent an email to all of them this morning saying, normally we use the susceptible, infected, removed, susceptible model, which means we group people into people that haven't had the disease, people that currently have the disease, people that have recovered from the disease. And if you've just had COVID-19, you're probably pretty safe. The immunity fades and the virus changes, but then they go back to the susceptible category. And the problem with that model is it assumes you know who's infected. And as the questioner notes, we don't know who's infected. So I proposed the susceptible, infected, detected, removed model, where we put into the model the knowledge that there's a whole time period before people become detectable after they're infected. So, you know, that's one way you can deal with it, is modify your modeling techniques to take into account this particular form of very bad ignorance. Yeah, number of patients in intensive care blocks is another pretty good measure. It's going to give you the same answer as deaths. There will be more people in those intensive care wards. The only problem is that in Italy, for instance, they overflowed the intensive care wards. So in Italy, the relationship between number of people in intensive care and number of dead people broke down. It sort of depends on how bad the country is. Over.
Yeah, and I think part of this is also, it's kind of the issues of comorbidities as well. Because Italy right now, it might not even be that somebody died of COVID-19. There's also the issue of people that are dying from complications caused by it.
Agreed. And in fact, there's another complication, which is that with COVID-19 people taking up all the space in the hospital, the guy who crashes his car can't get help and dies. And in fact, that's another thing you can do is, my God, don't do anything dangerous. No skydiving, no bungee jumping, no driving your car like a lunatic. I would also avoid getting drunk other than when at home and having no intention of going anywhere. You know, just stay safe. Now, I see that the questioner has added another question. What are the strategies for after quarantine time? Will the borders be closed for another year? Okay, I have no idea, but probably not. We're probably going to be to the end of the first wave by July, and maybe before that, depending on how fast it goes. And also depending on whether this coronavirus responds to hot weather and humidity by calming down. Now, annoyingly, the whole world is connected, and when it's summer in one half of the world, it's winter in the other half of the world, and so that causes a problem. And in the tropics, these diseases typically are around at a lower rate all the time. Having said that, we don't know what the good strategies are after things die down, and this will be, you know, something we're trying to do research on, and that individual governments are going to have to figure out for themselves. I think that limiting international travel is probably a really good idea. I'm guilty of way too much international travel. I left Korea 2 days before they had their COVID-19 spike. I then went to Australia, since I didn't want to fly across the Pacific Ocean twice, and I left Australia 3 days before they started having a lot of cases. It's like I've been walking out of houses just as they catch on fire. And then I missed Canada's "you must self-isolate for 2 weeks" directive by about 5 days when I got home. Now, I'm well past the quarantine period and I don't have symptoms. So I probably don't have the disease yet. But the IEEE, which Joey and I run or Dr. Brown and I run conferences for, is working very hard on virtual conferences. They're not having in-person conferences. Canada's National Research Agency has just said we may not use research dollars to travel outside of Canada until further notice. And in general, while the borders probably won't be closed, they'll probably be closed to anything not deemed really necessary. I hope that answers the question. Over.
Okay. So I think, oh, yep, that's a thank you is a response. Okay. Does anyone else have any questions for Dr. Ashlock while we have him here?
I have another set piece, if that's okay.
Sure, go right ahead.
So I spend a lot of time on social media, partly because it's the fastest way I know of to read 100 different news sources all at once. My friends and I have similar interests. They tell me things. I mean, I found out Boris Johnson had COVID-19 through Facebook, not through reading a newspaper or a newspaper's website. But one thing that keeps coming up that I've heard several times is, what the hell is flatten the curve? Because unfortunately, the epidemiologists talked about this and explained it once, and the popular media picked up the phrase because flatten the curve, it's flatten the curve, 3 words. It's a nice slogan. But apparently, a lot of people didn't know what it meant. Now, if you don't do anything, then the incidence of the disease rises very quickly, as in Italy, to where it overwhelms the health system. And then once almost everybody's had the disease and you have the harsh form of herd immunity, it dies back down. If you slow the spread of the disease, the number of people that get sick may be pretty similar to the number that would get sick if you let it spread like wildfire, but they spread out in time. So the number of people sick at any one time doesn't hit a very high level. In particular, hopefully it's below the maximum capacity of the healthcare system. And that's all flatten the curve means. Spread out the infection over time so that you don't overwhelm your hospitals and clinics and pharmacies and medical distributions. And Joey, you are recording this. I just got something about, sorry I'm late.
Yes, I am recording this, and I'll post it up and make it available for everybody past the meeting. So the software itself has a recorder inside of it, so it's going this entire time.
I'd also like to thank Dr. Brown for helping me hammer through and get Zoom running, because I'm on sabbatical this year, but the way things are going, I may well need to teach my courses next fall through Zoom, and now I have it working. Since we have, I assume, quite a few computer scientists in the audience, I wanted to tell you the last bug I found. My university bought me this nice headset and that webcam that's sitting on top of my mouse trackpad right now. And Joey and I, or Dr. Brown and I, were unable to get Zoom running until I discovered that the microphone had been shipped with its volume turned all the way to 0. This leads to another thing. If any of you start working with the problem of modeling the epidemic, or doing molecular simulations to try and prioritize possible cures for testing or anything, I've been working in bioinformatics, the application of computer science to biological data, mostly molecular biological data, for 25 years now. And it took me far too long to learn this lesson. I actually have a degree in computer science in spite of being a mathematician. And being a mathematician means that I have a very organized system in my head for debugging code. But what it took me far too long to learn is, when you're working with a biologist, you must also debug the data. I believe Dr. Brown has run headfirst into this problem during his career as well.
Yeah, I think that's pretty much every day that I spent within the automotive industry was mostly debugging people and how we did the collection than it was necessarily debugging the process.
To give a very simple example, the very first job I ever had was taking sequences from corn and automatically formatting the newly sequenced corn sequences for deposit in the NCBI's genetics bank, or GenBank, which is one of the largest repositories of sequence data in the world. And the biologist I was working with said, yes, look for this sequence of DNA. It's where the construct that we used to build the artificial plasmid started. And I couldn't find it. And I couldn't find it. And I couldn't find it. And then I went to the biologist and went, I don't think that's in there anywhere. And he used a tool called BLAST, the basic local alignment search tool, and blasted the sequences. And he said, yeah, that's because those are chicken sequences. Hang on. And it turned out that the first 96 sequences from which I had chosen my debugging data weren't corn sequences. They were the last 96 well plate from somebody else's sequencing run, and they were chicken sequences. And so I was looking for a construct that wasn't there in sequences from the wrong organism. Always debug the data as well.
So Dan, if people are interested in more of your work, where can they find you? Can they email you? I know that you have a online blog. Do you want to let everybody know where that is?
Sure. So if you just search on Occupy Math and Dan Ashlock, you'll find the blog. And the last two posts are about bacterial drug resistance and me gathering a lot of the information I've just given you into one place, if you want to. You're welcome to email me. Joey has my email address, and he can pass it out to you guys. Or pardon me, I get to call him Joey because I was his advisor, but he's actually Dr. Brown, and I'm trying to remember that. But you're also welcome to email me. I may or may not answer quickly depending on what else is going on. And if you find my homepage at the University of Guelph, there's a lot of my research papers there. You click research and ePrints, and I'm going to be putting up some more soon.
Okay.
If you want to help with epidemic modeling efforts, then filter yourselves through Dr. Brown, because he knows what sort of programmers might be useful to me. Over.
Okay. Do you got time for one more question before we close out the session?
Sure.
Okay. So there's recent news on research for AI for processing radiology to detect COVID. Is this a good idea or maybe too late for the patient?
So there I'm going to go, I haven't seen this, so I don't know. If by radiology you mean some sort of radio frequency imaging of the molecules, I suspect that that's actually a very good way to detect COVID very early on that uses a multi-million-dollar machine.
I think this is having to do with images of lungs. So detecting whether somebody's infected or not infected.
So yeah, no, if you already have a lung infection, that's a good way to confirm, yes, this is a patient, but that would be way too late for the patient to be helped much. I mean, for that matter, if the patient's getting their lung x-rayed, they're already in a hospital and probably already have a bed. I was thinking of using nuclear magnetic resonance to detect molecules unique to the virus. And in fact, that would work really well, except that right now, NMR machines are huge and expensive. We're about 3 years out from being able to use superconducting quantum interference devices to build handheld nuclear magnetic resonance machines. Unfortunately, that's 2 years and 11 months too late. Over.
Okay, so thank you all for joining.
Oh, and sorry, when I said nuclear magnetic resonance, I was using the old name, which is used for everything except medicine. In medicine, it's now called MRI because the word nuclear scares people, even though it's the same technique. I noticed the question was saying MRI. So yeah, MRI would be really good, but it's too expensive right now. Sorry, back to you, Joey.
Okay.
Dr. Brown.
So I guess the elephant in the room is that Dan actually was my doctoral supervisor. So he's going to continue to call me Joey for the rest of my career, which is perfectly within his right.
I could try and call you son.
So thank you very much again, Dr. Ashlock, for joining us today. And thank you to the participants who joined us tonight. I hope that all of you take away some interest from this and maybe some more understanding about what's happening right now. I will be posting this as soon as possible up on YouTube and making it available if you want to share it. And Dan, do you have any final words for everybody before you head off?
Follow the directions. Remember to wash your hands if you touched anything that might have virus on it, and stay home as much as you can. Stay safe and good luck.
Okay, and that message from me as well. Stay safe out there and please wash your hands.